Hello everyone, welcome to the virtual exhibit for E-Day. Today we are gonna represent the Biomedical Optical Imaging Lab directed by Dr. Yu. The Biomedical Optical Laboratory is located in the Department of Biomedical Engineering at University of Kentucky. This lab is mainly focused on the design and development of optical spectroscopy and imaging system for in vivo measurement of tissue hemodynamic and metabolism. Before getting started, let's talk a little bit about some basic but important concepts which can help you to understand what we are doing in our lab. Wavelength, as you can see here, can be defined as the distance between two successive crests or troughs of waves. It measured in direction of the wave. And on the other hand, frequency, as you can see here, is the number of waves that pass a fixed point in a unit of time. As frequency increases, the wavelength needs to get smaller. And now in the bottom figure, which shows electromagnetic spectrum, if you look on the top of this figure, you will see some sort of wavelength units. If you go toward the left, your wavelength increases, and if you go toward the right, your wavelength will decrease. So if you go toward the right here, you are getting toward the X-ray. And also, if you go toward the left here, you are getting toward the longer wavelength regions, like radio wave and the spectrum. In our lab, we mainly work and we are interested in the middle region, I mean near infrared region. Now let's talk a little bit about absorption and scattering. What we really mean by absorption or scattering of light through tissue or any other medium. Light can be transmitted through the medium and come out of the other end. Light transmittance, which can be calculated by this formula, is the ratio of transmitted light to the incident light, and the absorbance is the negative log of the transmittance. Which means that by increasing the absorbance, the amount of transmitted light will be decreased. And in this slide, in top figure, you can see this phenomenon. For example, when we go from right side to the left side, the absorbance is increased, but the amount of transmitted light, as you can see here, is decreased comparing with this one. On the other hand, the scattering can be caused by particles of different sizes inside the medium. It will be redirect light to any direction, as you can see in this figure. The light, the input light is redirected to many different directions because there are some scatter, some particles inside this medium. Now, what's the meaning of the light-tissue interaction? We want to know what will happen when we shine a light into a tissue. When shining a light into a tissue, photons will experience multiple events. The first things, as you can see here, is reflection. And it means that when we shine a light, it can sort of reflect off the top surface, which we are familiar with it. It's very similar to reflection of light from surface of a mirror. And the second option is remission. Which means that light can go through the tissue and most photons will be redirected to new direction by elastic scattering, and some lucky photons will eventually escape from the surface as reflectance after multiple scattering events. And the third one is absorption. As you can see here, it means that photons will be killed by some absorbers. And finally, which means transmission, and you can see it here, means that photons will go through the tissue and they will come out of the rare surface of the tissue. They either hit an absorber or they can just directly pass through the tissue and come out. So here is an absorption spectrum of all things found inside the tissue. This can be an animal or a human tissue. As you can see here in this graph, the black line here is water. Water is very broad and is very interesting for us because at this wavelength region, I mean between 300 nanometer to 1000 nanometer, its absorption is very low, and that's why water appears transparent when we look at it. There are other proteins and hemoglobin. Hemoglobin is a big part and it comes in two flavors, oxyhemoglobin and deoxyhemoglobin. And on the other hand, in the UV to near infrared wavelength range, the major scatters in tissue are cell nuclei, mitochondria, and collagen fiber with a size from 10 micron to submicron, which is close to the light wavelength. Photons are most strongly scattered by those structure whose size matches the photon's wavelength. Imagine you shine sunlight on a medium of interest and you measure whatever you can with whatever detectors you have, so you can make sense of what is inside the tissue. So for experiment measurement, then we will require a light source, like for example sunlight, or for example LED light or laser light. All of those will work for sort of imaging and a spectroscopy that we want to do. After this, you need to detect by some detector, and detector can be done by camera. In our lab, we use camera for detection. You need some sort of medium to couple the light from the source to the tissue and then pick that back up. And for this purpose, we can use the fiber or optical fiber because they are very convenient. Experimentally, when we are doing optical spectroscopy of tissue, we have some unknown. We want to see if the tissue healthy or it's a cancer tissue. But then, what do you do with this, for example, for this? Now you are getting measurements from a very complex medium that you have no knowledge about. What we are gonna do with those measurements? After finding some measurement and getting some data, we need to analyze these measurements using a specific theoretical model like diffusion or Monte Carlo model. The idea is that after getting these measurements, we have got our models. We can use our model and our measurement to get something useful that is the goal of tissue optics. So we want to quantify not only how much light reaches inside the medium or is coming back from it, but also we want to objectively determine the status of biological tissue using these measurements. We wanted to derive this optical property of a medium. This is a very hard problem, and it's actually called the inverse problem. On the other hand, in the forward model, which is easier to implement, we have some predefined value of optical property like scattering and absorption coefficient. Then we will get the simulated spectrum. Then we compare the simulated and experimental measurements, these two, iteratively until we get the best match with the lowest range of rate of error. Now, after learning the basic concept of biomedical optical imaging concept, now we are going to introduce some of our lab's activities and some of the main application of these concepts. The major application of optical instrument in our lab is 3D diffuse optical imaging of tumor hemodynamics and metabolism. And the next one is muscle hemodynamics response to exercise. And we also can mention cerebral hemodynamic monitoring. The main technique which is being used in this lab, in our lab, is speckle, contrast, diffuse, correlation tomography, and spectroscopy. It's called SCDCT. Then the underlying mechanism is the utilization of laser as the light source. All of us are familiar with sort of this pattern. That you see if, for example, you have a laser and when you point that laser to the wall or when you shine it toward the wall, the back reflected lights from the surface of the wall will interfere with each other. And this phenomenon is called the speckle pattern. The thing about the speckle pattern is that there are dark spots everywhere in the image and there are obviously bright spots. As well, if there is a moving object behind it, like for example, red blood cell in our body, you are gonna start seeing these speckle pattern start to fluctuate. The speckles will sort of blip, right? Those blips can be seen in this figure, here, which is the measured intensity versus time, those ups and downs. They will fluctuate in a pretty high frequency. If you measure this signal and you take the G1, here, if you can see, of the curve, which is basically normalized autocorrelation of that, what you get is the G1 curve. And that's what we measure here. So we are going to measure this curve by measuring these intensity fluctuations. The curve will decay faster if we have a fast flow and will be shifted toward left, and it will decay slower if we have a slow moving object. This is our measurement, and then we will fit it using model like diffusion model. Here we put a SC-DCT techniques. This is a speckle contrast diffuse correlation tomography technology. You can see it here, which has been recently developed in our laboratory. And this technique can be used for non-invasive three-dimensional measurement of tissue blood flow distribution. And here in the right figure, in the right hand side, you can see the schematic of the SC-DCT system for 3D imaging of tissue blood flow distribution. Here there is a galva mirror, which is actually it's a device. We can use it to deliver the point light to different positions. And also there are multiple pixel window. You can see it here, which can be used in order to detect a spatial speckle contrast at different location. And the coherent point source, like for example laser light, can be used to diffuse through a banana-shaped photon pathway inside a deep tissue volume and reach to the detector. You can see here there is a banana shape. This one is our source. It can be light from our laser, and this one is our detector. It can be something like camera. And also we have a line pass filter and a pair of polarizer, and the goal of these two parts is to for example to help us to reflect light directly from the source. And also we have a zoom lens, which can be used for adjusting the size of our region of interest. And here is a picture of 3D imaging of blood flow distribution using the SC-DCT, which has showed which has put it here to show you these techniques. In this figure, I mean in the part A, you can see the imaging setup, and in part B is you can see the reconstructed 3D distribution of blood flow SCDCT using SCDCT. The part and part C, you can see 2D cross-section views of flow distribution of different depths. And finally, at part D, you can see top view of blood perfusion measured by fluorescence and angiography with the injection of ICG. Here the dashed black box can show the fluorescent image indicated the region indicated in our region of interest, and the dashed I mean here dash red ellipses indicate the area with low blood perfusion observation, the fluorescent angiography. And here you can see the modified SC-DCT system for imaging of cell blood flow in a small animal like rat. In part A, you can see the experimental setup for reconstructed cerebral imaging of a rat. And in part B, you can see the optical design of the modified SCD-CT. As you can see in this part, there is the iris was used to limit the size of light spot. And finally, here is the rat head, and the hair of rat was shaved and removed with hair cream to expose a region of interest. And yeah, we have our region of interest is 20 by 20 millimeter, and we have a 5 by 5 source, I mean the circle here, and also we have a 20 by 21 by 21 detector. I mean the cross point here shows our detector. And here we are gonna to show you a video in order to have a better understanding of SCDCT and how it works. You can see in this video that here the light source is scanning the rat head by galvanometer. And finally, I'm going to say thank you for watching and listening to our virtual presentation. Hope you enjoyed it and have a better understanding of biomedical optical imaging and what we are doing in our lab. And I also would like to thank all of our funding agencies which provided us great financial support for all of our research projects. And as you can see, here is the name of our advisor, and here is our lab members. And this presentation is provided by Farane and Samane for E-Day 2021. Thank you.